All right, so continuing on with this idea of gene expression, um, again, to be sure, all of this stuff here, this is only referring to prokaryotic cells. Only the prokaryotic cells have the operon system where they organize genes that work together at the same position. There's a bunch of different ways eukaryotic cells can regulate genes, and a big reason for that is we have a lot more like organelles. We have the nucleus. We talked about how we can't do translation at the same time as transcription because the translation in eukaryotic cells happens in the cytoplasm. Transcription's in the nucleus. A similar thing can happen in gene expression. We have like dimmer switches, I guess I'm trying to say. We have a little bit more control perhaps with gene expression, a little more different ways to regulate than just a bacteria cell would. And this ability, and this is really important for eukaryotic cells because most eukaryotic cells are part of multicellular organisms, where multicellular organisms we have cell specialization, meaning we have like different cell types that make up tissues, tissues make up organs, organs make up organ systems. So we have a much bigger need to like make sure you have a specialized nerve cell, muscle cell, immune cell, and so on. And gene expression is really key in regulating all that, as we talked about before. It's worth pointing out that all cells have the same genome. They all have the same set of genes. The only difference is how they express, or what we call differential gene expression. So expressing genes, different cell types express genes at different levels and at different times. And like a breakdown in gene expression, a breakdown in, again, how often you use certain genes, that's a key thing that leads to a lot of different diseases like cancer. So understanding gene expression, like when I'm teaching, I didn't learn about this until I was like a junior in college. So the fact that they're teaching you this now in AP Bio, it's a sign that this is where we're really moving if we ever want to make big progress on things like cancer. I know this stuff can seem like it's a lot of details and kind of complicated, but like life's complicated, I don't know what to tell you, right? If we want to make progress on this stuff, we got to understand things at the genetic level. So, and I think it's cool. I think it's interesting kind of seeing how the complexity of some of these parts and stuff. So, all right, getting into it, these highlighted or colored in boxes, this is representing different levels that we can regulate gene expression in eukaryotic cells. So it's worth talking about and pointing out when we talked about like cell signaling that I mentioned how you typically have a signal that comes to a cell. The signal could bind to the outer part of the cell membrane. It could go in and travel to the nucleus. But that signal, I told you to think about that it changes gene expression. It turns on or off genes. So that's a way we can adjust gene expression. We talked about chromatin. That's how we package up DNA so it fits inside the nucleus. We can regulate gene expression by changing how which genes we unpack or pack up. We can then regulate genes at the transcription level, changing how often or how little we do transcription. A similar kind of idea we talked about with the operons with bacteria. We can do RNA processing. This is unique to eukaryotes, where we can change which part of the mRNA is an exon versus an intron. That changes different protein products. So that's a way we can alter the DNA. We can add the cap and the tail. That can change depending on like the adjustments to the cap and the tail. That can change how often we're going to do translation of that mRNA. We could then even degrade mRNA. Say like we have enough of the protein, we could just destroy all the mRNA that we've already made if it's really crucial that we stop making that protein for whatever reason. We could increase translation or decrease translation. We could change how we process that protein. That's a way we could adjust kind of how we use these genes. We could degrade protein. Say again, we don't need to use a certain enzyme quite as much. We could just destroy that protein. This is inefficient. You don't want to do that, right? It's more efficient to it's like there's a saying that an ounce of prevention is worth a pound of cure. It's easier to stop things like where they start at the gene expression level than to be like just destroying proteins, because that took resources to make those proteins. But that is a way that we could adjust if we need to kind of different gene expression and stuff. All right. So I said, yeah, it's easier to control things like ounce of prevention, right? Control things where they start at transcription. So that's where we commonly are going to control gene expression. And again, the complexity of eukaryotic cells, that's what gives us a lot of these options that we just went over. Okay. All right, I added this slide in from your book. This slide, if you get this slide, you actually understand really most of the rest I want to talk about today. And this is epigenetics, which you talked about a little bit. Epigenetics is basically controlling how often you increase or decrease gene expression. But the simplest way to think about it. And to understand epigenetics, you need to remember that to fit DNA inside the cell, we wrap it around these things called histones. Okay? And if you have the DNA that's like super wound up around a histone, we call that heterochromatin. I'll write that over here actually. So heterochromatin. So heterochromatin. That is DNA that's tightly wound around those histones. And heterochromatin is a decrease in gene expression, GE. Okay? And when you think heterochromatin, I want you to think of methylation. The attachment of methyl groups, which is what you see right here, these methyl groups, attaching these methyl groups to histones, for whatever reason, you don't worry about the chemistry of it, it will cause us to not use, that's what I'm trying to show here, this is DNA inaccessible. We can't access that gene. How is RNA polymerase going to get to that gene if it's wound around those histones? Okay, that's what we call the heterochromatin. Okay, I blocked this out because they kind of mistaken. They said they said also you could activate it. Really, methylation, that might technically be true in some rare scenarios, but methylation, when you think of that, think it decreases gene expression. Now, why this is important is because I mentioned it last class. Like there can be certain chemicals you could be exposed to, or just like by living a certain lifestyle, that will change how often, like these chemical adjustments we make to the histones. Meaning we'll end up changing how often we use or don't use certain genes. And it's a super important idea. Changing how often, like how we methylate certain genes, that can make a big influence in like how you can even like pass down certain like traits to like offspring. Like they found like as I mentioned last class, like certain like pregnant mothers, like if they ingest certain chemicals and stuff, that could change the methylation patterns of that developing embryo, which could then lead to like maybe an increase in maybe in something like diabetes or certain mental health disorders. A lot of the details around this are still kind of being worked out, but anyways, the main idea is actually fairly simple. Adding these chemical groups will allow us to access or not access certain genes. Okay, so moving on. A lot of this kind of already said, but let me make sure I've said everything. This second bullet point is kind of a more fancy way of saying it, right? The promoter of a gene relative to the nucleosomes. The nucleosomes, that's a collection of 8 histones. It's a nucleosome. So that gene promoter, if it has a bunch of methyl groups on it, that gene promoter is tightly wound in the heterochromatin formation around all those nucleosomes, and you can't access that gene to do gene transcription, to end up making that protein. Okay. Now, you may have been wondering, well, cool, heterochromatin, that's when we decrease gene expression. So then how do we increase gene expression? And I mentioned this word a little while ago, but euchromatin. So euchromatin is when DNA is unwound, for lack of a better word. It's not tightly wound. So it says this is to be like your euchromatin right here. The DNA is not as tightly wound around those histones. So then this would cause an increase in gene expression. And I want you to know if methylation caused heterochromatin, what will cause your euchromatin is acetylation. Here's how you can remember that. I used to like playing poker when I was like your age, and like DNA is unwound here. We use the word open. probably a better word, and tightly wound, closed. So in poker, you typically want to wait around until you get a good hand before you play it. And everybody knows, or hopefully you know, that the ace, that's like your best card. So you are open for business, like you want to be doing gene expression, you want to play that hand in poker when you got the ace. When you got the acetyl groups bound to the histones, that unwinds the DNA, and now the promoter and the rest of that DNA region is open, where we can then do gene transcription and use those genes and make the proteins. So euchromatin, and it's good, right? It's good, it's open. Now heterochromatin, methylation. Meth is bad. Don't do meth, right? You do meth, just decrease in gene expression. It's about the best I got for you. So, anyways, any questions about this? I think it's a fairly simple concept. Okay, send everything here. Again, acetylation. Attached to amino acids and histone tails. Again, the details of why acetylation opens up genes, not really as important for your purposes right now. This last bullet point, not as important, but I think it's worth pointing out that there are some other chemical groups we can attach to those histones, and then that can then we talked about these protein complexes where one protein binds another protein binds another protein. So you can have something like that can also happen, like a chemical group attracts another as a binding site for another enzyme that can then further modify the chromatin. But the end result is ultimately you increase or you decrease the gene expression. You open up the chromatin or you condense that chromatin more. Okay. DNA methylation, adding the methyl groups. Yeah, the more heavily methylated genes are, the less likely they are to be expressed. It's kind of interesting. Once you methylate certain genes, it tends to remain like that throughout different cell divisions. So getting back to like public health and risk for it. So if you're exposed to certain chemicals, the worry is you're going to methylate certain genes, and then you keep passing on that methylated genes to different anytime any of the cells in your body do cell division, and that's an issue for you and your own personal health. But think about it if it's the gametes, whenever you're actually making those gametes, it's been shown that those methylations can then be passed on down and be inherited by other offspring. Okay, details of that are still kind of up in the air on how exactly that works or which chemicals cause that, but that's a big area of research. If you're kind of interested in this, that's kind of something maybe in college or something, you try to find a professor that does kind of some research on that. Same thing can be passed on to future generations. We call this epigenetic inheritance. Epi means above. It's above genetics. So it's like changes in gene expression, changes in how those genes are used that don't involve the nucleotide sequence. Doesn't involve the actual sequence of A, T, C's, and G's. Okay. In other words, we talked about mutations last class. Where mutations, you actually change the nucleotide sequence. That would change whether the protein works or doesn't work. Here, we're just changing how often we use the genes. Okay, so it's important to make that distinction. And the benefit of this is we can reverse these, right? So that's kind of the hopeful thing with this. We can folic acid is I don't know if it's a vitamin a lot of pregnant mothers will take. That will help change the methylation patterns of the histones, and that can help reduce some of those negative side effects. So there are some ways we know we can try to reverse some of this, whereas mutations more or less get locked in. So mutations are maybe a little bit scarier in some ways than epigenetic modifications, depending on how you view it. All right, any questions? Last slide. All right, cool.